But there will be time after her presentation for questions, and you can either unmute yourself if you'd like to chime in for those questions, or if you look kind of in the toolbar, there's a little show conversation chat box, and feel free to write your question in there and post it to the chat box. I'll monitor that, and I can, I guess, say your question for you if you don't either have a microphone or don't want to say it out loud. The other thing you can do if you have a question is there's kind of a little emoji button, and you can raise your hand. So if, like I just did, if you have a question at the end of the presentation, feel free to raise your hand, and I'll try to go through kind of the people with their hands raised and allow you the time to answer your questions. Hopefully that makes sense to everybody. After 10, 15, 20 minutes of questions with all of you folks, the committee will kind of get back together with Cadie, and then we'll kind of take her through the process of asking her the questions that we have. So, and then we can all get on with our days. I think that's all on, the format side. So I'm excited to introduce Cadie Olson, who's a fellow pointer who graduated in 2018 from UWSP. She worked in the aquatic biomonitoring lab at UWSP, where I think she gained a lot of experience with Jeff Dimmick, identifying benthic invertebrates in streams. After graduation, she moved on to the Minnesota Pollution Control Agency, where she worked for a while, and then started her graduate degree or graduate work here at UWGB in 2019, which she'll obviously be talking about. Cadie's moving on to bigger and better things down in Missouri. I believe it's the Department of Conservation down in Missouri. She's got a full-time gig down there as a aquatic macroinvertebrate biologist. So congratulations to her. And we're really excited about her work, and I think it's going to be something that'll be referenced by a lot of people for kind of a long time to come. So take it away, Cadie.
Thanks for the introduction, Chris. I'm really excited to share my last 2 years of work with you guys. This was a really fun project, and I'll dive right into it, the macroinvertebrate community of Green Bay. But before I get started here, this research really would not have been possible without the help of others. And so first and foremost, I would like to thank my funding sources, the Wisconsin Sea Grant and the Wisconsin DNR and EPA AOC funding. Big thanks to my advisors, Dr. Chris Houghton and Dr. Patrick Forsyth, for their support and guidance throughout this project, as well as Dr. Stahlheber for her statistical guidance. Really couldn't have done it without you guys. So thank you. Kurt, Jeff, and Kara have devoted countless hours to teaching me taxonomy and helping me with sample processing and identifications. And I really can't express enough gratitude for that, so thank you. The technicians and interns with the Aquatic Ecology and Fisheries Laboratory spent a ton of time helping out with field work and lab tasks that made all of this possible. If you guys don't already know that you're amazing, you guys are amazing. And then last but not least, big thanks to Bruce Markert for giving me a baseline study to go off of and his taxonomy assistance, as well as Dr. Lisa Grubisha for lending me her microscope throughout these past 2 years. And so with that, a little bit about my study area. My study was focused in southern Green Bay. And Green Bay, even though it's part of Lake Michigan, has some pretty unique attributes that distinguish it from other areas within the Great Lakes. Green Bay's southernmost tributary, the Fox River, is characterized by agriculture and industry and has been throughout the last century or so. Historically, paper mills discharged polychlorinated biphenyls, or PCBs, into the Fox River that were integrated into sediments and living things. And then on top of all that, nutrients and sediments from upstream are transported downstream and into Green Bay. And I know a lot of you guys are familiar with Green Bay and the Fox River, but for those of you that aren't, the Fox River alone supplies 70% of Green Bay's annual sediment load and about 1/3 of the annual phosphorus load to all of Lake Michigan. And that's crazy. That's a lot. So this creates a concentration of nutrients and sediment near the mouth that lessens northward as the Fox River's influence is diluted. And we call this phenomenon the trophic gradient. And so nutrients, especially phosphorus, fuel primary production in algal blooms, just like the ones shown in these pictures. And sometimes these blooms are clearly visible in satellite imagery, like this bottom right photo. And then the ones in the bottom left here are from more recently. This was some of our field work at UWGB in 2019 on the Fox River. And all of this plankton and algae dies eventually, which contributes to a really high sediment oxygen demand as they decompose. And decomposition, coupled with seasonal stratification, really depletes benthic oxygen and leads to regular hypoxia in Green Bay. And so this dead zone of low or no oxygen usually establishes in late summer off the east shore near Dykesville, for those of you locals that know where that is, which is shown in red on this map. But there are a lot of other mechanisms that are at play that complicate hypoxia even further. So southerly winds and current circulations drive cold water from Lake Michigan into Green Bay, which encourages this rapid stratification. And as the water moves south from all of the primary production, it's depleted of oxygen. And so in short, wind direction, currents can really affect the extent, duration, and location of the dead zone. And so the mobility of hypoxia has been pretty well studied. And this figure here is from Klump and his co-authors in 2018, and it shows how this dead zone can shift. So hypoxia is represented in red in this map, and as you can see, it deviates from that east shore location quite often. For example, even taking up the entire width of the bay in September of 2012. And PCB contamination, the trophic gradient, and hypoxia are just a few of the legacies from agriculture and urbanization. But as many of you know, these issues are not new, and they started to be addressed with the Clean Water Act in the 1970s. And a lot of improved water quality measures kind of fell out from the Clean Water Act, including the enactment of a lot of total maximum daily loads for nutrients, or TMDLs, the reductions of point sources of nutrients and pollution, and the designation of the Lower Green Bay and Fox River Area of Concern, or AOC, which is this area off to the right here on the map. And since the designation of the Area of Concern in the 1980s, many of the PCBs have been removed from the Fox River through dredging, which, if you guys pay attention to the news, that concluded last spring. And additionally, the Cat Island chain was restored in the 2010s after it was washed away by storms in the 70s. And efforts like these have really earned Green Bay the title of a proving ground for restoration. And after years and years and years of degradation, it's finally in an age of improving water quality. And we can trace these trends in water quality with things like chemical measurements and habitat assessments, but personally, not that I'm biased at all, I really prefer the living things because they integrate all of these attributes of their environments. So benthic macroinvertebrates are good candidates to assess water quality because, unlike fish, they're a little bit more sessile and they're longer lived than other organisms like plankton. And macroinvertebrates have varying tolerances to pollution and nutrient enrichment, sedimentation, oxygen levels, depending on the species. So generally, good water quality indicators are pretty sensitive to things like low oxygen and high sedimentation. And some of the organisms that are sensitive to those things in Green Bay I have shown here on the left, labeled in green. So those include things like mayflies, scuds or amphipods, native mussels, unionid mussels, and sowbugs and isopods. On the other end of the spectrum, there are some poor water quality indicators, and those include things like oligochaete worms and chironomids. However, there are exceptions to every group, so some midges and worms are considered indicative of healthy environments, and so taking the time for lower level taxonomy can be pretty valuable. And macroinvertebrates have been examined in Green Bay in the past, and the communities do shift over time to reflect these changes in water quality. And so these comprehensive surveys began in about 1938 and occurred roughly every decade after that until 1978, but the sampling methodology did vary. So when I talk about this graph, just take that with a grain of salt. And I do want to mention too that other surveys have occurred since 1978, but they weren't quite as extensive as the efforts listed here. And the you'll learn that I'm about to compare my study to the one from 1978, and that study references these studies as well. So I just chose them for consistency. And so this graph off to the right shows the abundances of major taxonomic groups from Serber and Cooley's survey in spring of 1952, Haumiller and Beaton survey in spring of 1969, and Markert's survey from the summer of 1978. And the data shown are from 22 sites that were sampled in each one of those surveys, and average abundances are in meters squared, and they're shown with an X. And so looking at this top graph here, worms are shown in light blue, and they drastically decrease from 1952 to 1978. And again, those are one of those poor indicator taxa, so it's that's a positive water quality trend to see a decline in their abundance. Midges, shown in dark blue on the top, have slightly increased over time, and then the middle and bottom graphs are showing the abundances of several other a little bit more sensitive taxa, and for some reason, the abundances of amphipods and isopods absolutely skyrocketed in 1978. But others, like fingernail clams and leeches, have remained pretty steady over time. And isopods and amphipods are good water quality indicators, so these trends are interpreted as positive changes and signify that the water quality has likely improved over time. But a broad assessment of Green Bay's bug community really hasn't been conducted since 1978, and so some knowledge gaps are pretty apparent. An updated macroinvertebrate inventory is needed to assess the effectiveness of recent restorative efforts and political measures. A lot has happened since 1978. And although the dynamics of eutrophication and periodic hypoxia have been pretty well examined, not much is known about how those things affect bug communities. So I attempted to address these knowledge gaps with my study. And I had 3 objectives. So my first was to describe the current macroinvertebrate community of Green Bay and quantify the magnitude of change since 1978. My second objective was to assess the influences of productivity, substrate type, and depth on macroinvertebrate communities. And then third, I wanted to quantify the role that hypoxia plays on macroinvertebrate assemblages. And so to answer my objectives, I took 258 samples around Green Bay. 97 of those sites were historical stations that were also sampled by Bruce Markert in 1978, and those are shown in white in the left-hand map. 100 sites were added to increase spatial resolution, and those are the black dots on the left. And all 197 sites on that left-hand map were sampled in June and July of either 2019 or 2020. And then later in summer, in July through September, I surveyed during hypoxia at the sites shown on the right. We took a total of 61 samples, which included 14 samples taken at 5 benthic oxygen sensor locations, which are those white triangles. And I addressed the effects of hypoxia on benthic communities using several different methods, so I'll take the time and break apart that sampling regime a little bit more later. But I do want to take a moment and talk about the regions I have listed on that right-hand map, because I do refer to them quite a bit. So the inner bay is analogous with the bay portion of the area of concern, and it's located south of Longtail Point and Point Sable. The area above that is the lower bay, and it extends from Longtail Point to the entrance lighthouse. And I don't have the entrance lighthouse on this map, but it's about where Littletail Point is. And then the region above that, from the entrance lighthouse to Sturgeon Bay, is the middle bay. And Sturgeon Bay is outside of the scope of this map, so my study didn't encompass all of the middle bay. And then just so you guys are aware, there is a 4th region called the upper bay that goes all the way from Sturgeon Bay to Little Bay to Knock, but I didn't look at that, so you won't hear about that one. And so all the samples were collected with a Ponar dredge, which is what Brandon is pulling up in this video. It scoops up sediment from the bottom, which we then empty into a sieve that sifts out most of the debris, but retains the macroinvertebrates. And the remaining material was put into jars, just like what Aaron is holding on the right, and it was preserved with formalin and rose bangle stain. And the rose bangle stain allows us to better see macroinvertebrates when we're picking them out of debris back in the lab later. And so that's why we do that. At each of these sites, we also collected depth and oxygen readings, as well as hand-textured substrate. And all these samples went back to our labs to be processed. Just thought I would give you guys a little bit of a look of what our lab looked like last summer in the midst of COVID. So we did all of our lab operations in our boat shed, and there were some challenges like no running water, but we were able to social distance and spend some time outside, which I thought was pretty fun. But looking at every single bug and every single one of these samples would have taken forever. So we subsampled with a device called a Catan tray, which is just a gridded method that we would take a random grid and then pick grids to completion until a representative subsample was obtained. Worms and chironomids comprise the majority of samples, and lower level identification of those taxa requires visibility of specific anatomies under a compound microscope. So we mounted a lot of those on the microscope slides, like that middle picture there. And then finally, all of the subsampled macroinvertebrates were identified to the lowest level, which was usually genus or species. And if you guys were curious, this is what some of our invertebrates looked like under the scope. A lot of bugs were involved in this project. So just to put things into perspective for you guys, of the 258 samples, Green Bay did 178. And then one of my committee members, Kurt Schmoody, did 80 of them at Lake Superior Research Institute up in Superior, Wisconsin. But just considering UWGB, we identified about 20,000 macroinvertebrates, and we estimated that the processing, mounting, and identification processes took about 1,200 hours, which would equate to 52 days or 31 work weeks. So again, I just really want to drive home how awesome our AEFL team was these past 2 years. And so after all the sample processing and identifications were finished, I could finally start to address my objectives. And so I first described the current macroinvertebrate community of Green Bay and then compared it to historical conditions. I use predictions and results from some previous studies to kind of preface my own. Two particular taxa, the tubificid worm Limnogilus hoffmeisteri and the midge chironomus, were consistently dominant over time, and I anticipated that they would continue to be. But because of recent restoration efforts and habitat improvements that I mentioned before, I expected that the community would have changed for the better since Bruce Markert's survey in 1978. I kind of expected the abundances of those happy face green bugs, the isopods and amphipods, would continue to increase. But I also expected spatial distributions of taxa to shift in response to specific habitat changes, like the restoration of that Cat Island chain, which kind of created a more wetland-like hydrology in that specific location. So I thought the bugs would respond to that, as well as shift overall just from broad-scale nutrient implementation efforts that broad-scale nutrient control efforts that have been implemented since 1978. So my survey found 95 unique taxa, and most of those were worms and midges. Most of the worms belong to the widespread subfamily Tubificinae, but there was quite a few Cnidinae present too. And together, worms and midges made up about 91% of Green Bay's benthic community based on abundance. So this pie chart here, the 60.97% is the tubificid worms. The cnidid worms made up about 1.5%, and then the chironomids made up about 28%. Worms averaged around 5,600 per meter squared, but they were collected in abundances over 100,000 per meter squared. And worm abundances that high are usually symptomatic of severe nutrient enrichment. We collected worms from all late summer samples, or all samples except one, which was one of the late summer ones during hypoxia. Midges were a little bit less widespread, but still pretty much found everywhere. We got them at 249 sites. They averaged about 2,600 per meter squared in abundance, and they topped out around 29,000. And even though worms and midges were dominant, other groups were pretty common, including dracidid mussels, which are zebra and quaggas, and fingernail clams, which are listed as Spheridae, and snails, gastropods. And as I predicted, Chironomus and Limnogillis hofmeisteri were the dominant identifiable taxa. But I was curious how these taxa distributed in Green Bay, so I mapped some of the common ones. These maps were created with inverse distance weighted, or IDW, interpolation, which is just a method that puts weight on known values and averages the values in between known ones. So starting with these bottom two maps here, Darrow and Arcdionaeus lamandi were the most abundant nidid worms. And even though nidids only comprised about 1.5% of the community, Green Bay had some pretty decent worm diversity, so I just wanted to see where these were located. So the highest concentrations of Darrow were found at one site off the east shore near Dykesville. And then Arcdionaeus lamandi was found more over by Littletail Point and by the Swamico River mouth on the west shore. Limnogillis hofmeisteri, the top right map, was our most common worm, and it was really abundant in the eastern lower bay, again near Dykesville. And if you remember me talking about hypoxia earlier, that's right where it commonly establishes. So I'll preface that for later. Chironomus is extremely abundant in the inner and lower bays, but if you guys notice, the scale only goes up to about 1,200. And so I broadened it out a little bit to show where the highest densities of Chironomus actually occurred. So Chironomus was concentrated in the southwestern inner bay, near the mouth of Duck Creek, and near the mouth of the Swamico River. And at those locations, their abundances exceeded 10,000 midges per meter squared. You guys will continue to see maps in this fashion throughout the rest of the presentation, and they're all going to be made the same way with IDW interpolation. So I wondered if these healthy trends would continue over time, and this is the graph I showed you earlier with my data tacked on at the end of it. So again, this is just for 22 sites that were sampled in each survey, and averages are shown with an X. And so midges, which are the dark blue boxes at the top, continued their increasing trend. And then worms, the light blue boxes at the top, remained pretty similar to 1978 rather than decreasing, which is what I anticipated. But they were more variable, as you can tell by the width of the box. Also, kind of contrary to my hypothesis, the more sensitive taxa didn't change all that much, or they decreased. So looking at the bottom graph, amphipod populations, which are the dark blue, crashed since 1978, and so did isopods. It appears that 1978 was an anomalous year for those two taxa. But I was most interested in comparing my results to the most recent survey in 1978. So if I consider only the 97 sites that were sampled in both my survey and 1978, there were some differences present even after I adjusted for taxonomic name changes. So my data is shown on the left, and I found 68 unique taxa at these 97 sites. And those included 30 oligochaete worms and 13 chironomids. But at the same sites in 1978, richness was a bit lower, and Markert identified 59 taxa. And that included 28 worms and 14 chironomids. And 39 taxa were represented in both surveys, but 50 were only collected during one time period. So 20 taxa were collected in 1978, but I didn't collect any in my survey, and I collected 30 taxa that weren't collected in 1978, which is pretty interesting. But if we consider both surveys and the unique taxa associated with both of them, 89 taxa have existed in Green Bay at some point. I further summarized these shifts since 1978 using metrics, which can also provide some helpful insights about community health. And so I calculated all the metrics listed here for my data and 1978. Abundances you guys have seen so far have been in organisms per meter squared, and you'll continue to see them in that way. I also determined richness, Shannon diversity, and dominance at the genus level. And for diversity and richness, I included only unique taxa, and so that excluded immature worms, which did comprise a big component of the samples. I assessed dominance with DOM1 and DOM3 metrics, and DOM1 just means the percent of the sample that the most abundant taxon comprised, and then DOM3 is the percent of the sample that the three most abundant taxa comprised. On top of that, I calculated a modified trophic index, or MTI, for each historical and contemporary community. And so the index just summarizes trophic condition based of trophic condition of a location based on tolerance values. So taxa are assigned tolerance values ranging from 0 to 3, with 0 meaning that taxon is tolerant of oligotrophic conditions, and 3 means it's tolerant of very eutrophic conditions. And these tolerance values are then incorporated into an equation that spits out a score that also ranges from 0 to 3, with scores less than 0.6 indicating that the macroinvertebrate community is suggesting that location is oligotrophic, and then scores between 0.6 and 1 meaning mesotrophic, and scores greater than 1 indicating eutrophic conditions. And I added on to this index just a little bit by enumerating the abundances of the eutrophic taxa, so those that were designated a 3, and the combined abundances of mesotrophic and oligotrophic taxa, so those that were assigned a 1 or a 2. And I called those abundtal and abundintal, respectively. I use statistical comparisons of means and medians to test if several of these metrics have changed significantly since 1978. I use 2 sample t-tests assuming equal variances when assumptions were met. Otherwise, I square root transform data, or I use Mood's median test. I did consider the other non-parametric equivalents of a t-test, the Kruskal-Wallis, but my data didn't meet assumptions, so I just used Mood's, which is slightly less powerful. All the analyses you're going to see for this and other things use an alpha of 0.05. And starting off with mean Shannon diversity here. Mean Shannon diversity has decreased since 1978. And this table off to the right is showing means with standard deviations in parentheses. So Shannon diversity decreased from 1.72 to 1.42. And we can examine these maps to see where these differences actually occurred. And interestingly, Shannon diversity now appears to be the lowest in the southern Middle Bay, where it was once the highest in 1978. And I suspected that this would be due to dominance, which is considered in Shannon diversity calculations. And so I looked at that next. And mean dominance by a single genus has increased since 1978. And this increase is pretty clear on maps. The contemporary map on the left is much darker, which represents higher dominance. And it really appears to have increased near the mouth of the Swamico River, which is kind of interesting because it wasn't very high there in 1978. I then kind of wondered if worms and midges were contributing to higher dominance. Median worm abundance was similar across my survey and 1978. But again, just as a reminder, this table is showing means. So the means reflect this phenomenon as well, but I didn't test those. But again, I just absolutely love these spatial comparisons because even though there isn't statistical significance, you can see that their distributions have changed quite a bit. So worms were once pretty localized, kind of over by the mouth of Duck Creek in the Inner Bay, and shifted to elsewhere in my survey. Median chironomid abundance, though, has increased since 1978 from about 1,300 to about 2,400. And mapping revealed expansions of midges into the inner and western lower bays. To answer how communities have changed in composition since 1978, I use NMDS, which stands for non-metric multidimensional scaling. And this is an ordination technique that portrays communities as points on a defined number of axes, and that's what this plot is on the right here. Just to heads up, I'm going to spend a little bit more time on this slide, just because I use these analyses to address my other objectives. So points represent communities, and the proximity of points matters. So points that are very close together mean that those two communities are very similar. And on the other hand, 2 points that are very far apart represent 2 very different communities. The yellow points on this graph are showing the communities from 97 sites that were sampled in my survey, and the gray points are showing the same 97 sites from 1978. The centers of each group are shown with ellipses, and we call these group centers centroids, which are basically spatial averages. And you can compare those spatial averages with an ordination equivalent of an ANOVA called a permanova. And so the centroids look pretty far apart here. They're not, the ellipses represent confidence intervals, 95% confidence intervals, and they're not overlapping at all. And the permanova indicated that the separation of centroids is significant, which just means that average community structure in my survey is different than average community structure in 1978. One downfall of a permanova is that it doesn't always separate differences in variance from differences in centroids. So in other words, points could be really spread out in one group and really clustered in another, but the spatial averages could still be different. So to test variability among communities in each group, I used a dispersion analysis. And here dispersion was significant, indicating that new communities were more clustered or more similar to each other than historical ones. And now, I guess, what do these arrows mean? So these arrows are vectors, and the lengths of the arrows matter. So longer arrows means that that taxon was more important, and the arrows can also be kind of thought of like pseudo-axes or regression lines. So the closer points or a community it lies to a vector, the more influence that that taxon had on that site. So you may notice that Chironomus, Green Bay's most dominant midge, is a really long vector that extends through many of the yellow contemporary communities. So basically, that's just showing that Chironomus is a bigger deal in my samples than it was in historical ones. And then flip that around, all the other taxa are kind of pointing down toward the historical sites, so those were more influential there. I kind of validated these vectors using a species indicator analysis, which is just used to determine if certain taxa were more associated with one survey more than the other. And the species indicator analysis complemented the NMDS vectors very well and concluded that the isopod Cassidoidea, which is one of the vectors, the amphipod Gammarus, which is another one of the vectors, and the midge Tannitarsis were associated with 1978 communities. So as an amateur statistician, that just made me extremely happy. And so the NMDS basically determined that the communities are different, but I wanted to know if these changes were uniform or if things have changed a ton in places and not as much in others. So each contemporary community on the NMDS corresponded to a historical one in the same sampling location, and I took the distance that corresponding communities were apart in NMDS space and I mapped them. So again, points that were farther apart on the NMDS were more different, so greater distances just indicates more change. And so red is showing communities that changed the most since 1978, and that seems to be up north in the Middle Bay. And the Inner Bay, though, seemed to remain pretty similar to 1978 with smaller Bray-Curtis distances. And so I determined that communities were different from 1978, as I hypothesized, but differences in habitat can cause differences in community structures too. So for my second objective, I assessed the influences of productivity, substrate type, and depth on benthic communities. So productivity and depth have been shown to drive benthic community structures in other Great Lakes, but Green Bay is pretty shallow, and the Fox River is known to contribute a significant nutrient load to the ecosystem. So I expected that productivity and the trophic gradient would drive community compositions more than depth and substrate type. I also expected that eutrophic communities and silk communities would be more characterized by tolerant midges and worms like Limnogilus hoffmeisteri and Chironomus. But first, I thought it would be helpful to see what these habitat types look like in Green Bay. So depth is the map farthest to the left, and depth follows a latitudinal and longitudinal gradient in Green Bay. So it's generally shallower near the mouth of the Fox River, and it gets deeper as you go north. And then the west shore is pretty shallow, and the east shore is deeper. But overall, southern Green Bay, my study area, was really shallow and didn't exceed 20 meters in depth. And substrate, which is the middle map, also followed a longitudinal gradient. The west shore was mostly sand and the east shore was mostly silt, and other substrate types like mussel beds and clay were kind of sporadically sprinkled in there. I used the modified trophic index for the right-hand map as kind of a surrogate for productivity. So this map is showing that communities are responding to the Fox River's trophic gradient. And so MTI scores were high, representing eutrophic conditions in the inner bay near the mouth of the Fox River, and kind of faded to mesotrophic with decreasing scores as you went north. And so I started off by summarizing the influence of some of these abiotic factors with NMDS and vector overlays again. And so the points shown on this NMDS are representing the 197 sites that I sampled in June and July. And the vectors can be interpreted just like the taxa vectors you just saw. So depth and longitude have shorter arrows, which means they were less important, which makes sense because depth was pretty homogeneous in Green Bay, relatively speaking to other Great Lakes. Latitude and MTI, though, are really prevalent arrows, which suggests that productivity and the trophic gradient have the largest influence on community structures. This is the exact same ordination that I just showed you. Dots are all in the same place, but I separated them by region to illustrate the effects of latitude and the trophic gradient a little bit more. So this tells the same story as the latitude vector and the MTI map from earlier. So inner bay communities are represented by light green dots. Lower bay communities are represented in dark green, and the middle bay is blue. A permanova indicated that these three centroids differed, which means community structures in these three regions were different from each other. The dispersion analysis, though, showed that the variation among these three groups also differed. And so I explored these specific regional differences and community variability with Tukey's on significant differences. So inner bay communities and lower bay communities were significantly more similar to each other than communities in the middle bay, which you can kind of see in this plot here. So inner bay and lower bay communities, the green dots, intermingle quite a bit, which just means that these communities likely aren't distinguishable from each other. The middle bay, though, is a lot more spread out. The community structure was a lot more variable. I did the same thing to look at substrate type, but I specifically assessed sand and silt and tried to determine if those two substrate types affected community compositions. And so this ordination might look a little bit different because I only included the sites that were sand and silt, and there was 160 of them. I excluded the other substrate types just because I had some sand with silt and silt with sand designations that just would have made things confusing. But what this is showing is that communities are different in these two substrate types. So sand communities were more variable and silt communities were a lot more clustered. So communities that lived in silt had much more a similar composition than those in sand. And then again, I used a species indicator analysis to kind of identify if any taxa were going to be associated with one substrate type or another. And it turns out that Dracaena, Spherium, and then the worms Isocades, Podomothrix, and Spirosperma, and the snail Valvata were more likely to be found in sand, which kind of surprised me because community structure is more similar in silt. I expected the associations to be more with silt than sand, but that's how the analysis worked out. So that was pretty interesting. And so depth, productivity, and substrate type are players in other ecosystems, but regular periodic hypoxia is a pretty unique aspect of Green Bay that complicates things. And so I wanted to explore how these how hypoxia affects benthic assemblages in Green Bay because it hasn't been explored in detail yet. And so I did so using 3 different methods that consider dissolved oxygen concentrations at the time of sample, as well as the duration of hypoxia and the frequency of hypoxia. Overall, I hypothesized that hypoxia would limit the benthos. I expected differences in community structures would be most evident when considering chronic hypoxia, so hypoxic frequency and duration. I didn't really expect benthic fauna to respond all that much to differences in dissolved oxygen at the time of sample because the hypoxic zone is so mobile. So because much of the bay is exposed to these stressful spurts of no oxygen or low oxygen, I expected biota to be pretty universally tolerant. And so the first of my 3 hypoxia analyses considered dissolved oxygen at the time of sample. And for this analysis, I used 61 samples that were collected in late summer of 2019 and 2020 when hypoxia was ongoing. I resampled stations when possible, so these 61 samples encompass 36 locations. My sampling events were triggered by benthic dissolved oxygen conditions less than 2 milligrams per liter at the northernmost buoy, so the northernmost triangle on this map. I checked benthic oxygen levels in 2-kilometer increments from that buoy in each cardinal direction with a sonde. So I would stick the sonde in the water north, south, east, west, and then once I found oxygenated conditions, I took a sample there, and then I backtracked and took another one in hypoxic conditions. Most of the samples were taken in that fashion, but 14 of the 61 were taken at these 5 benthic sensor locations, again, which are the white triangles on that map. And so to assess if communities differed based on hypoxic condition at the time of sample, I grouped them as hypoxic, so less than or equal to 2 milligrams per liter when I collected them, or oxygenated, so greater than 2 milligrams per liter when I collected them. And I visualized that with an NMDS. And so this is what that looked like. And this might look a little bit different than the other ones I've shown you because no real patterns are evident. So the red communities that were collected during hypoxia mingle a lot with the gray points that were oxygenated at the time of collection. And the centers and dispersions of these groups weren't all that different, which suggests that macroinvertebrate communities subjected to hypoxia at the time of sample were not distinguishable from those that were oxygenated. But I also looked at hypoxic duration using the 14 samples collected at the 5 sensor sites. Since these sensors were constantly collecting oxygen data, I could determine how long each specific location was hypoxic for. And so sensors ended up being hypoxic anywhere from 1 to 22 days, and that's cumulatively. So it wasn't hypoxic for 22 days all at one time. That was throughout the course of the season. I chose an arbitrary cutoff of 8 days to use as groupings for an NMDS to visualize if communities differ structurally when they're subjected to longer durations of hypoxia. And so 3 of these 5 sensors were hypoxic for 8 or more days, and I took 10 samples at those 3 locations. And then on the other hand, 2 buoys were hypoxic for less than 8 days, and I took 4 samples there. And so this is that ordination. The analysis was a little bit less powerful just because few samples were involved, only 14. But the trends that this is showing could warrant some further exploration, and I think someone should go out there and put out a couple more sensors and take macroinvertebrate samples at those locations to see if this trend continues. But even though only 4 samples were part of the short duration group that was less than 8 days hypoxic, a Perminova still deemed these centroids different, which means that the communities exposed to hypoxia for more than 8 days were different from those exposed to hypoxia for less than 8 days. But the dispersion analysis came back as insignificant, so communities were variable regardless of their hypoxic condition. And then again, I did an indicator analysis to see if certain taxa were associated with greater than 8 days of hypoxia, and they were. Chironomus and Limnodrilus were. So that was kind of cool to see because, again, thinking back to those maps from earlier, they occurred most often in the highest abundances where hypoxia most often occurs. The third and final method of examining the effects of hypoxia on benthic macroinvertebrate communities considered the frequency of hypoxia. And this one is personally my favorite. I think it's the most interesting. So I showed you guys this image before, and I used the maps from this figure from Klump and his co-authors in 2018 in combination with my data to map where hypoxia has occurred most often from 2009 to now. So In this instance, I switched things up a bit, and I considered hypoxia to be 4.5 milligrams per liter or less. And recent studies have suggested that the traditional threshold of 2 milligrams per liter is too low, and living things are often affected before that. So some other studies have examined cutoffs around 4 milligrams per liter, so I just chose 4.5. And so I digitized these areas subjected to less than 4.5 milligrams per liter from 2009 to 2015, and then 2019 to 2020 in ArcGIS Pro. And so I had this map of a bunch of overlapping polygons, and then I used a frequency account overlapping polygons function to determine where hypoxia occurred the most often. And so that made a map that looked like this. So red is showing where hypoxia less than 4.5 milligrams per liter occurs the most, which I considered polygon overlap of 8 to 11 times, so the reddish orange color. And I bet you guys can see where this is going. These are the last of my NMDS ordinations, I promise. I separated the early summer samples by frequency of hypoxia, and I visualized them with NMDS. So all the sites that are within this orangish reddish color, I deemed frequently hypoxic, and the sites outside of that I considered infrequently hypoxic. And I chose the 197 early summer samples rather than the 61 late summer samples, just to have a bit bigger of a sample size, so I had more samples within the hypoxic, frequently hypoxic area. But macroinvertebrates also fluctuate seasonally in their community structures and abundances, so I left out the 61 late summer samples just for temporal consistency. And so this NMDS is showing that communities subjected to frequent hypoxia differ from those that were not. So PermaNova suggested that these centroids differ, again, meaning that the community structures are different between these two groups. Dispersion was also different, as you can see by the clustering of the red frequent communities. And this just means that communities exposed to frequent hypoxia were pretty similar to each other, and the ones outside of frequent hypoxia had a lot more variability. And just like some of the NMDSs I've shown you before, I overlaid taxa vectors. And so immature tubiphysses without hairs, Limnogillis and Chironomus, were more influential on communities exposed to frequent hypoxia. And then conversely, the snails and mussels pointing down were more associated with infrequent hypoxia. I did a little bit different of an indicator analysis for this part that looked at species combinations rather than just single taxons. So instead of saying, is this taxon associated with one group or the other, it looks at combinations. So this analysis identified the nided worm Archdionaeus lamandi in combinations with Spherium, Procladius, and Chironomus and immature worms to be more associated with frequent hypoxia. And so my findings can be used to assess the health of the Southern Green Bay ecosystem. But the moral of the story that I think my study showed is that this community is really like no other. So other Great Lakes ecosystems are typically more dominated by dracinid mussels, and they don't have a worm presence as extreme of Green Bay's all the time. Overall, the community here reflects a very eutrophic, nutrient-rich environment. And standalone, the community appears poor. But because we have all these awesome historical studies to compare things to, we can see that things have improved since 1978 and since other surveys. So increased prevalence of chironomids is a positive change since Bruce Markert's study in 1978, even though chironomid abundance did contribute to higher dominance and lower diversity metrics, which are typically considered negative attributes of ecosystem health. Benthic community dynamics and metrics varied regionally because of the Fox River's trophic gradient. And because each region functions differently, it's pretty challenging to compare Green Bay to other ecosystems, and each region should really be assessed individually. And so if we do that and we look at each region by itself, the inner bay, so the AOC portion of Green Bay, appears to have improved since 1978, which is signified by higher richness and decreased worm dominance. But the lower bay may have degraded a little bit. The MTI scores indicated increased nutrient enrichment, worm abundance has increased and diversity has decreased, but this may be due to hypoxia, which from my hypoxic frequency map commonly establishes in the northeastern lower bay. The middle bay is a little bit more confusing. So the MTI scores suggested that nutrient enrichment has decreased since 1978, but richness has also declined. And it's typically like, yay, decrease in nutrients is good, but we don't really want to see species dropping off because of that. But it is pretty typical for mesotrophic and oligotrophic communities to have lower species richness than eutrophic ones. But it's I think it's quite challenging to kind of determine if that's what's happening, or again, if hypoxia is at play. And hypoxia is kind of limiting what organisms can live in certain regions of the middle bay. And so several measures can be taken to address degrading communities, maintain the improving ones that we're seeing. And one of those is phosphorus abatement, which has been ongoing for a while now. So I think that education and advertisement of best management practices should continue. Continued monitoring of benthic communities is needed to assess how macroinvertebrates have responded to the Fox River dredging. So even though my study caught the responses from the tail end of dredging, it takes years for communities to acclimate to these new conditions and recover to a disturbance as extreme as a giant vacuum sucking up your home. So it would also, like I mentioned before, be interesting to explore if community structures differ with longer durations of hypoxia using more samples and sensor locations, since my study only had 14 samples. And again, my study did not examine the upper bay, so that again, that's the area north of Sturgeon Bay, and a new inventory is needed there. So that would be a cool project. Quantifying the health of Green Bay is pretty challenging because the dominant taxa are worms and midges, which are very seasonal. So kind of establishing a threshold of the community is healthy if we're under 20,000 worms per meter squared is hard because they fluctuate so much. But if I think a good objective to work toward would be this decreasing trend in worms complemented by an increasing trend in midges. So even though at the beginning of the presentation, I showed both of those as kind of poor indicator taxa, midges are typically considered a bit better than worms. Again, identifying a model ecosystem could be challenging because of apparent regional differences in Green Bay, but I think it would be a good place to start. And Lake Winnebago has been thrown around as a good candidate for the Interbay, and it just might be. And so here are the wonderful references that I referred to throughout this presentation. And with that, I'll take any questions. That was a lot in a short amount of time.
Well, I'll start. I don't have a question, but I will say thank you, Katie, because I, prior to this presentation, never really understood how to read NMDS graphs. And now I feel like I do. So you totally taught me something.
Good to hear.
Are we able to ask questions orally?
Yes, of course.
Okay. This is John Luchai. I have two questions.
Sure.
One question was, you went back to the 1978 locations and sampled those in addition to a whole bunch of other locations. Because they didn't have GPS back then, how were they able to locate their sampling sites, and how were you able to find them again?
Yeah, that was a challenge for sure. The 1978 study did have coordinates, so I went off of those. But before that, it was all hand-drawn maps. So I haven't talked to Bruce Marker to see how he chose those sites and resampled them. but I'm sure there is some margin of error. I know the other studies have mentioned, the older studies pre-1978 have mentioned accuracy within 100 meters, which to me is a lot. That's quite a bit margin of error. There can be a lot of changes in microhabitat from 100 meters.
Yeah.
But that was one of the reasons why we added so many extra sites, just to make sure that we weren't missing
Sure.
any of those small microhabitats.
Yeah. Well, I mean, that's just a limitation of technology, right?
Mm-hmm.
The other question I had for you is, it seemed like, well, your images of the organisms were fantastic and beautiful. I really like the microscopy part of it.
Oh, thanks. I have to mention, I did a couple of those, but most of them were Kara Kamke, one of our technicians here. She was a frickin' rock star and loved doing it, so I let her. And made for good presentation images. So thanks, Kara.
So my question is, because you guys have so much maybe easier access to color, imagery, books, and things that I'm not sure what was available in the 70s. I went to college in the late 80s and early 90s, and I can tell you just the difference in being able to have stuff on the internet, look it up. Things have probably changed a lot in 40 years. Do you think that some of the differences in the species that you found could be differences in either what you could identify, how things were lumped or split, or maybe not just changes in the taxa in how they were named, but maybe even some that hadn't been discovered, or any other variables, like they maybe didn't know and so they lumped them in with a different species?
Yeah, that's definitely a possibility. And I was actually in direct contact with Bruce Markert, thanks to one of my other committee members, Jeff Dimick. And so I did ask him about that. And there was a couple that came into question that we addressed. But the other taxonomists on my committee, Kurt and Jeff, could probably attest to that a little bit more, because I'm not as familiar with how the names have changed over time and how groups have been combined, given that I've only been doing this for like 5 or 6 years. But yeah, it's definitely a possibility.
Okay. Thank you.
Yeah. Oh yeah, so I see Jacob just messaged. Can you go over again the taxa that increased and decreased with the persistent hypoxia and put common names on those? Yeah, let me shift back to that. So the longer durations of hypoxia, we had Chironomus, which was our most common midge, and Limnodrillis, which was our most common worm, a tubificid worm. Were more associated with longer durations of hypoxia. And then if we, maybe go back to this one. So again, ITWOH just stands for immature tubificid worms without hairs. So those were comprised a large component of our samples, because lots of worms can't be identified. We need to see certain reproductive anatomies for a lot of them in order to identify things. And so those, and then again, the same taxa, Limnodrillis and Chironomus, were more associated with more frequent hypoxia in this case. Does that answer your question? And Bree asked, Lake Winnebago has significant nutrient issues as well. Is this a good reference because it doesn't have the same legacy sediment contamination issues as the Fox and Green Bay, and also doesn't have hypoxic condition to the degree that Green Bay does? All good points. And I have brought up the Winnebago as a reference site for the Interbay to other folks as well, and that's also a point they argue quite often. And I agree. I don't know as much about Lake Winnebago, but I know for reference sites, usually geographic proximity is an important consideration. You want a similar climate, precipitation, all that stuff to the site of interest. And Lake Winnebago, I just knew was extremely eutrophic, has the blue-green algae problem that we do, and is generally really shallow, just like the inner bay of Green Bay. So that is one of the closest ones that popped into my brain. Other Great Lakes also popped in my head, like Lake Erie, which is also shallow, eutrophic, has very similar issues. But again, looking at Lake Erie's macroinvertebrate community, the little bit of literature I've read about it is just way more dominated by dracinid mussels, specifically quagga mussels, and less so dominated by worms, which I found really interesting. And I think Klump in one of his papers mentioned that the reason Green Bay might not have the dracinid abundance as other Great Lakes is because of the fluidized sediment that Green Bay is known to have. So Green Bay doesn't really have a hard bottom. There's like this floating layer of this fluid silt that kind of floats around that isn't very conducive to dracinid establishment. And then Dr. Luchai, do you think the exceptional precipitation of 2019 might have had an effect on the study? Yeah, probably. And so I'm glad we really sprinkled out my sampling sites between 2 years. And I did do like a couple exploratory analyses, and 2019 and 2020 weren't different in their community compositions. The NMDS, when I did that, which I didn't include in here, unfortunately, looked a lot like this one. So the communities mingled from 2019 and 2020 a lot, which suggests to me that maybe the community structures weren't all that different between those 2 years, despite the high precipitation. But I'm sure it affected it a little bit. Mm-hmm.
Hi, Katie. This is Paul Baumgart. Can I ask
Hi.
a question early?
Of course.
Would higher lake levels and high bay levels make a difference, say between 1978 and 2019 and 2020?
Yeah, I mean, it definitely could have. I know most of the surveying efforts between all the studies were more focused on profundal habitats than littoral ones. I would expect, just based on some other stuff I've read, that littoral benthic communities would be affected a lot more by those fluctuating water levels, just because a lot of the taxa that live there are reliant on wave action, or if there's coarse substrate, a lot of that too, rocky shorelines and stuff. So I'm not sure. I'm sure it would have some effect, but I'm not sure it would be an extreme degree, just because a lot of the organisms that we're sampling are just burrowers that live in silt.
Thanks.
Mm-hmm. Oh, and then I see, Chris, could you suggest any possible mitigation strategies? Do you mean for phosphorus or nutrient abatement? Chris?
Yeah, I guess I'm wondering, is there anything that you can actually do with the sediment to mitigate? So rather than just relying on changing water chemistry, and is there something more drastic that could be done, possibly?
Yeah, I've thought about that quite a bit. It's really hard because nutrient abatement and sediment abatement really rely on public cooperation, and a lot of us scientists point fingers at the farmers, which I'm sure there's some scientific validity to that, but I think the best thing we can do is the encouragement of those best management practices. Areas that are susceptible to erosion, put in silt fences, practice no-till, practice cover crops, windbreaks, all of those things. I actually wrote a little bit about that, I think, for one of my finals for Dr. Stahlheber's classes. And I probably talked about those things exactly. Oh, and then Mike, based on your knowledge of the literature, are there any known biological interactions between or among these taxa that might influence the population dynamics of these animals? Certainly. I think the most well studied is probably dracinid mussels. So lots of certain kinds of macroinvertebrates prefer different substrate types, especially coarse substrates can attract a lot more diversity. And so dracinid mussels, whether they're dead mussel beds or live druces, they offer that, even if it's kind of artificial because it's a living thing. They offer this coarse substrate, all these little spaces for certain organisms to hide from predation. And then they also produce pseudofeces that offer food for a lot of collector-gatherer organisms, particularly things like amphipods and isopods. So those tend to be a little bit more abundant in locations where dracinids are present. And then there's also been a study or two on Lake Erie that looked at hexagenia. And hexagenia was also found to be more abundant in some places on dracinid beds, just as a mechanism to hide from predation, even though they're a burrowing mayfly. So to me, that was super backward, but very fascinating. Oh, Dr. Lucci again. Were any samples kept or sent for stable isotope analysis to establish baseline 15N values, etc., for trophic level studies? Yes, they were. If Brandon is still on here, maybe you could talk about that because your research is way over my head. But yes, he did go out and collect some macroinvertebrate samples for his study as well that were sent to somewhere in California to be analyzed. So he will be talking about that in a few months at his defense, I hope. Oh, Jake, is it possible to do restoration of the aquatic plant communities on the bottom? That is a great question. Again, I'm kind of thinking back. I worked really closely with Brandon Fallish, who is the other graduate student on my grant that's doing stable isotope analysis. And so he sampled the macrophyte communities quite a bit. And if I remember right, it's a lot of milfoil and something else. Yeah, but I'm not really sure how you'd go about restoring that. That's it. Thank you, Sago. Yes.
I guess just a quick comment would be that it's really dependent again on nutrients and water clarity. So we can address some of the issue. You can even replant, put in plugs and stuff, but until maybe some other issues are addressed, those mitigation strategies might not be that effective. All right, I think the questions are starting to trail off here. Maybe we'll give it a couple more minutes if anybody was a little shy in asking their questions. But I think we'll go on to the our chat with the committee members. And Cadie, any last words?
No, but if there's any burning questions, I realize I didn't put my email on this slide, so I'll stick it in the chat. So if you think of anything later, feel free to shoot me an email and I will answer. I'll do my best to answer.